We'll just wait for 1-2 minutes and then we'll start the session. Is is my voice audible? Okay, so a very good evening to everybody and happy Janmashtami to people who are celebrating. So today we'll be discussing questions for week 5. So we'll be discussing questions which are related to week 5 course content. Okay. So in week 5, yes, thank you, Ajay. Yeah. So we'll be discussing about week 5 questions. So now that we have understood about the differential gene expression, we have also understood basics of genetics, which is involved or mostly used in the developmental biology. Now our concern is understanding some kinds of cell-to-cell communications. Okay, so now we want to see and learn about cell-to-cell communications. Cell-to-cell communication plays a very important role when we are studying about developmental biology because many kinds of signals are received by certain cells, and those signals, upon receiving the signals, the cells start performing in a certain way. So the cells, if they are not induced by such signals, they will not perform in the way they have to, so which is known as induction. So induction means a phenomena where a particular cell or a system, which is known as inducer, is secreting some signals which are received by another tissue or another group of cells, which is known as a responder. Okay, so you have an inducer and you have a responder. So if I say I have a inducer, mm, I don't know where is my, just a minute. Huh. I have an inducer and I have a responder. Okay, so what is this both makes the induction process. Okay, this induction is very important in developmental biology. Now what happens over here is that suppose this is a cell and this is another area. Now this secretes some signals. Okay, and these signals are received by another cell. So this particular cell, which is or tissue, which is secreting some signal, is your inducer, and the cells which are responding to these signals, they are known as responder. And this whole process is known as induction. Okay, this whole phenomena is known as induction. Now what is important is that this kind of signal secretion and receiving can be of various kinds. If you have gone through the lectures, then you must have studied that it can be paracrine signaling or just juxtacrine signaling and all those things. So they can be of various types. I'm not going into detail because I'm assuming that you must have gone through the lectures. So this is just like why cell to cell communication is important, because for the development in any organism, the organism cells receives and takes some signals. So that is why some cell to cell signaling plays a very important role in that case. With that, we'll start with our first question. So our first question is, in a tissue recombinant experiment, which of the following will show lens induction? Okay, so genotype of the optic vesicle, these are the genotype of the optic vesicle, and these are the genotype of the surface ectoderm. Fine. So if the genotype of the optic vesicle is wild type and surface ectoderm pack 6 null, pack 6 null in both cases, or wild type, pack 6 null in optic vesicle and pack 6 null in surface ectoderm also, or wild type in both cases. So we have to tell, you have to tell that in which of the following combination eye lens induction would be shown. Okay, so you have 2 minutes to solve this question. After 2 minutes, I will switch the screen. Think and answer. Okay, so shall I switch the screen? Okay, I'm switching the screen now. Okay, let us see. So Ajay has answered B and D. So just a minute. Hmm. Hmm. So here the question says, in a tissue recombination experiment, which of the following will show eye lens induction? So here we are talking about, just as I discussed, that here we are talking about the phenomena of induction. So when I am saying here the phenomena of induction is taking place, that means induction, that means there has to be what? There has to be one inducer and there has to be one responder. Okay, and in this case, if you have gone through the lectures, you must know that the optic vesicle is your inducer and the ectoderm, surface ectoderm, is your responder. Okay, so now when both inducer and responder are of wild type, then it is very obvious that Ilan's induction will take place. So D is for sure short the correct answer. First thing. Now here it says if both are mutant, will there be any induction? No. So C cannot be the answer. Now these are the two important things. If your uh, inducer is mutant but responder is wild type, can there be a induction? Yes. Why there can be an induction? Because some basal level of induction it can show even if the inducer is not there. But if the responder is only not working, no matter how much induction is taking how much secretion is taking place from the inducer, there would not be any response. So A cannot be the correct answer. So the correct answers are B and D. So just as we have discussed that induction is a phenomena which requires two components. One is your inducer and second one is your responder. Just a minute. Hmm. So just as I discussed that your induction requires two components, inducer and responder. Inducer is the one that secretes the signal and responder is the one that responds, that gives response to that particular signal, that receives those signal. So in this question it is about eye lens induction where your optic vesicle is your inducer and your surface ectoderm is your responder. So the correct answer would be B and D. Now this is important, which I have taken from third's lecture only. So inducer is the tissue that produces the signal that changes the cellular behavior of other tissue. Okay, this is what inducer means. Then what is responder? Responder is the tissue that is being induced, that is receiving the signal, that receives the signal. Okay, so inducer produces the signal and responder it receives the signal. And here if you could see in the question, we are talking the question talked about pac6. Okay, so now what is the relationship of pac6 and with islands induction? So pac6 is an essential factor in anterior ectoderm to be able to respond to optic vesicle. That means this ectoderm, the response of the ectoderm to the optic vesicle is dependent upon PAX6. Okay, so if PAX6 is not there, then the if PAX6 is not present in your surface ectoderm, then it is unable to respond to the signal which is given by the inducer. So therefore PAC6 is an essential factor in the anterior ectoderm. Important here to understand is that it is an essential factor in anterior ectoderm. That means if PAC6 is deleted from the ectoderm, then there would be a problem. Okay, because PAC6 is important for anterior ectoderm and not for optic vesicle. That is why in this case, uh, um, we took marked B and not A. Do you understand this point? So now here is saying, now this says that PAC6 is essential for the anterior ectoderm to be able to work. So now if you see here, if both now here optic vesicle is your what? It is your inducer. Optic vesicle is your inducer and surface ectoderm is your responder. If both are wild type, then yes, there would be lens induction. If optic vesicle is PAX6 null. Now, what does this mean? How to read this? PAX6. This means PAX6 is not there. It is knocked out. PAX6 is knocked out. And why it is written two times? That means it is a homozygous knockout. That means in both the alleles, PAX6 is not present. PAX6 is absent in both the alleles. Okay, because we know that a gene has two alleles. So if it is written like this, it means that in both the alleles, your PAC6 is not present. It is a homozygous condition. What could have been in heterozygous condition? A heterozygous condition could be PAX6 is and wild type. That means in one of the allele PAX6 is absent, but in other allele wild type. That means PAX6 is present. Now what will happen? Will there be an islands induction? Yes. Why? Because PAX6 is important in surface ectoderm. Okay. If it is not present in surface ectoderm, then there would be no islands induction. It has not much importance in optic vesicle. Now, optic vesicle, it is wild type, but in surface ectoderm, PAC6 is absent. Then there will be there any islands induction? No. Why? Because PAC6 is essential in anterior ectoderm to be able to respond to optic vesicle. And if both are uh, in both the inducer and responder, in both the cases, if your PAC6 is absent, then obviously there would be no islands induction, because in surface ectoderm also PAC6 is absent. So this is the question about eye lens induction. It talked about the concept of inducer and responder, and it talked about the concept of relation of PAC6 with the surface ectoderm or the ectoderm. Now we come to the next question. The optic cup give rise to lens, retina, optic vesicle, or both the lens and optic vesicle. What should be the correct answer? 2 minutes to think, and then I will switch the screen. Hmm. Think what can be the answer. After 1 minute, I will switch the screen. Okay. Okay, I'm switching the screen now. Hmm. Hmm. So Ajay has answered as B, which is retina. Hmm. And B is the correct answer. So the optic curve, it gave rise to retina. So basically you need to understand that optic curve is a structure in the developing eye that give rise to retina, which is the light sensitive layer at the back of the eye. Okay. And what does your lens do? So lens forms the lens play coat. Okay. And not directly from the optic curve. It is formed from the lens play coat. And optic vesicle, it is an earlier developmental stage that eventually forms the optic curve. So just nothing much to explain here. Just you need to understand. Just a minute. Yeah. So Nothing much to explain over here. Just you need to understand that optic curve, it can give rise to retina because lens, what is how the lens is formed? Lens forms from the lens placode, from the lens placode, and optic vesicle. Optic vesicle is an earlier developmental stage that eventually forms the optic curve. So the correct answer is retina. Okay, fine. Now we come to the next question. Which final structures will be generated when dermal mesenchyme from the thigh and wing are recombined with the epidermal epithelium of the wing? Options are A. Wing feathers and claws, B. Thigh feathers and wing feathers, C. Scales, and D. Claws. Okay, 2 minutes. Think and try to answer this question. You have 2 minutes to think and answer the question. If you have gone through the lectures, then it is very easy for you to answer. Okay. I'm switching the screen now. Hmm. Okay. So Ajay has answered it as B, which is thigh and wing feathers, and Jasrina has also answered it as B, which is thigh and wing feathers. Okay. So Sangeeta, as you just joined, so this was the question that we were discussing, that which final structures will be generated when dermal mesenchyme from the thigh and wing are recombined with the epidermal epithelium of the wing. And the options are wing feathers and claws, thigh feathers and wing feathers, and scales and claws. So just Reena and Ajay has answered it as B. Now let us see what is the correct answer. The correct answer is B, that is thigh feathers and wing feathers. Now, how to explain this question? So, you have to understand that when dermal mesenchyme from different regions, okay, it any region, like here it is thigh and wing. So, when dermal mesenchyme from any different region is recombined with the epithelium, uh, uh, epidermal epithelium of the wing, then the epidermis usually forms structures which are appropriate to the region of the mesenchyme. Could you understand? So, final structures will be feathers corresponding to the source of the mesenchyme, which is thighs and wings. To explain it more correctly, see here, this was the experiment. This is the image which is taken from the your course. This is the concept of regional specificity of induction. Okay, it is a concept which explains regional specificity of induction. So you can see this is the source of dermal mesenchyme and this is the wing epidermal epithelium. So in all the cases it is same only. This is same. This is your epidermal epithelium only. But depending on the source of your mesenchyme, your induction is taking place. Okay. See, in each and every case, it is same wing epidermal epithelium. But if the source of dermal mesenchyme is wing, it will form wing feather. If the source is thigh, it will form thigh feather. And if the source is foot, it will form scales and claws. So this explains the regional specificity of induction in the example. Okay. Now we'll move to the next question. If the RTK pathway is compromised in C. elegans, defects in which of the following will be readily observable during larval development? A. Cell movement, B. Vulva formation, C. Locomotion, and D. Reproduction. 2 minutes to answer this question. One minute more. Think and answer. Okay, now I'm switching the screen. Hmm. Let's see. Okay. So Ajay has answered B and Justina has also answered B. And Sangeeta, do you also want to participate? What do you think should be the answer? Hmm? Think what should be the answer? No? Okay, fine. So the question says if the RTK pathway is compromised in C. elegans, defects in which of the following would be readily observed? So basically, uh, RTK pathway in C. elegans, it is responsible for vulva formation. Very easy question, huh? It's a very easy, simple question. You should be able to answer it. So basically, what is RTK pathway? RTK is your receptor tyrosine pathway. Okay, and it is very important during vulva formation during larval development, and any kind of defects which are observed in this pathway, it often leads to abnormalities in vulva development. Okay, such as failure to form vulva properly. So if there is any defect in the RTK pathway, that is receptor tyrosine kinase pathway, then there is problem or abnormalities in the vulva development, and your vulva is not formed properly. You can have a vulva-less phenotype also. Fine. Now, one thing that I wanted to discuss here, I think I must have downloaded the, mm, just a minute. Mm, I think it should be there. Hmm. Lecture 13, and I want lecture 14 also. Yeah. So in lecture 14, in the last slide, mm, yeah, here. Here you can see that whenever we are studying about developmental biology, so like any kind of biology that you are studying, whether it is your biology of the cells, cell interactions like developmental biology, or you are studying the biology or physiology of plants, or you are studying about animal physiology, so there are certain various pathways which are involved, okay? And the function of those pathways is to carry out the communication properly. So here in the case of developmental biology also, there are four major pathways which are very much important. In fact, if you are studying about developmental biology, you need to have a basic idea about these pathways. The first one is the fibroblast growth factor, or your RTK pathway, hedgehog pathway, your wingless pathway, or your TG and your TGF beta pathway. So these are the four important pathways, okay, that you need to learn or understand in developmental biology. So if you see here, it's like a basic. You should understand the basic of how a pathway works. Okay. Then you can see this is your RTK pathway, receptor tyrosine kinase pathway that we are studying. See, the role of RTK pathway in various species. In C. elegans, it is responsible for vulva development. In Drosophila, it is responsible for eye development. In human, it is responsible for cell proliferation. Why I am saying so? To learn and to read about these things, because these pathways are important not only for your exam point of view, but for in general if you are studying about developmental biology. And because if the question was asked about C. elegans, there might be a chance that they can ask about the role of RTK pathway in Drosophila or human. So you should know this. So this is basically the description of the RTK pathway. But what I wanted to explain over here, I thought of explaining you, is the basics of how a pathway works. Okay. Now, why I am telling you that you should know the basic of how a pathway works? Because see, whatever factors or whatever pathway we are studying about, whether it's a hedgehog, wingless, or TGF beta pathway, the ligands might change, the downstream molecules might change, but the concept remains the same. Okay, and if you are able to understand this concept of how generally a pathway works, then it is very helpful for you not only in developmental biology subject, but in general if you are studying about cell biology also. So always remember that when we are talking about such kinds of paracrine signalings, or basically we are talking about paracrine signalings here. So what happens is that there is a receptor, okay, and there is a ligand. The ligand comes and it binds to the receptor, okay, and when the ligand comes and binds to the receptor gets activated. Now, what is the meaning of the receptor getting activated is, if you can see here, this is the receptor. Now, the receptor has two sides. One is the cytoplasmic site and other one is the extracellular site. The extracellular site is the site which interacts with your ligand. And the cytoplasmic site is the site which is present in the cytoplasm. And these cytoplasmic sites have tyrosine kinase domain. Okay, they have tyrosine kinase domains. So whenever any receptor binds with the ligand, which is basically a paracrine factor, then these tyrosine kinase domains get activated. Okay. Now, what is the meaning of getting activated in the case of cell biology is that they get phosphorylated. So, phosphorylation is equals to activation in this case. So when they get phosphorylated, then they activate their subsequent downstream molecules. For example, you can see here when this got phosphorylated with the help of ATP, it activated another protein. And then this protein will activate some other proteins, and in this way a protein cascade would be formed and something at the end would be activated. So whenever you are studying about any kind of paracrine signaling, which is mostly involved in your cell developmental biology, uh, okay, this is the other question. Always remember, sorry, hmm, always remember that a question or something. Yes, Ajay. What is a ligand? Okay, so ligand is also a protein. Ligand is something that comes and bind to the receptor. Okay, so always remember everything is like whatever extra like I told you that inducer it secretes some extracellular signal, and that extracellular signal is received by the responder. So there, what is the nature of biology of that extracellular signal? That is actually some proteins only. So ligands are nothing but some proteins which are secreted by some of this one cell which is an inducer, and it received by some other cell which is responder. Now after the receival of that signal, some kind of pathway is induced in that tissue. Okay, and what is that pathway? That can be either RTK pathway or TGF beta pathway or your Wnt pathway. So there can be any pathway, like the four pathways that we just discussed. So ligand is essentially a protein that binds to the receptor. Is it clear? I hope it is clear to you, Ajay. So what I'm trying to tell here is it is a basics of paracrine signaling. Okay, if why I'm giving stress on this topic so much, because this will be clear
No,
for
madam, please, legend, I am yet to understand.
is it an antigen? No. See, it is not an antigen antibody interaction. Um, okay. So, one thing you should understand that these signaling molecules, this ligand which is secreted, this is not an antigen antibody interaction because your receptor is not an antibody. Your antibody is essentially your B cells, which is present on your immune cells. But that is a different concept. So you have a antibody. This is your antibody. Okay. This is the heavy chain. This is the light chain. And you have some regions where your antigen comes and binds. This is your antigen and this is your antibody. And after the binding of this antigen to the antibody, whatever signaling has to be done, I mean, whatever further downstream reactions has to be done, it is done. But this is not an antigen antibody reaction. But everything is protein. Everything is protein in the sense that transcription factors, what are those? These are also proteins only. So ultimately, the expressed part, whatever is the expressed part in the body, that is in the form of proteins. So antigens can be proteinaceous also, it can be non-proteinaceous also. It is not like antigens are always proteinaceous. No, your virus, is it a protein? It is not a protein. It has a protein capsid, that is a different thing, that is a bacteriophage. But what I'm trying to say that your pathogen can be a protein, it cannot be a protein also. Okay, but in general, pathogen is a protein because most of the pathogens have a proteinaceous nature only. But here we are not talking about antigen antibody interaction. Here we are talking about pathways, cellular pathways. Okay, cellular pathways are different from antigen antibody interaction. And if you want to see, yes, antigen can be a protein, antigen cannot be a protein also. In general, antigen is a protein and it binds to antibody. And the structure of antibody is different. Okay, antibody structure is different and receptors are different. Those are two very different things. Antigen antibody interactions are seen in immune cells. We talk about B cells. Whereas these receptors are not on the these receptors are, these pathways are generally involved in developmental processes. Okay. Like embryonic development or even B cell development also, but in the development processes, they are mostly involved. Antigen antibody interactions are seen after the development is completed when the cell is completely matured. Okay. Now, if we want to get an example, so this is the example. This RTK pathway is an example. This is the pathway in which your ligand binds. Now, what is the ligand? What can be the ligand that you have to see? I don't remember the names of all the ligands, but the ligands can be anything. Ligands can be your hormones also. Your hormones can also be your ligands. So, when your hormones bind to the receptor, it initiates a cascade. See, it's a very basic in a general concept that whatever reactions are happening in our body, they are happening through a pathway. Okay, they cannot happen without a pathway. There has to be a pathway, there has to be a signaling cascade that goes on and that causes some kind of function. Okay, just a ligand coming and binding to the receptor, then what next? There has to be something, some cascade has to go on after that so that it can do its function. So here we are essentially talking about paracrine signaling. So in paracrine signaling, you have a receptor, you have a ligand. If you have studied a little bit biochemistry, then you must have known what is a ligand, and you have a receptor. Okay, this is your receptor, and this is your cell membrane. This is your cell membrane. So this part of the receptor is the cytosolic part. This is your cytosolic part. And in this cytosolic part, you have tyrosine kinase domains. Now, these tyrosine kinase domains are activated only when a ligand comes and binds to it. And what is the meaning of activation? That means that they get phosphorylated. So these tyrosine kinase domains get phosphorylated. In cell biology, activation means phosphorylation. Whenever it is written they are activated, it means they are phosphorylated. So when they are activated, in in turn, they activate some other protein is activated, and then this activated protein will activate some other protein, this will activate some other protein. So it is a signaling cascade. One by one, each and every one is activating the other, just like a relay race. What is a relay race? You have something in your hand, you hold it, you run, and then you give it to the other person. So similarly in here also, this phosphorylation, this is activated, this will activate its further downstream molecule. After this activation, it will activate its further downstream molecules, and laterally in the end, what will happen that some in the last, whatever the thing has to be induced, it is induced. Like in this case, you can see ultimately um, your transcription factor is activated and it causes the expression. So our ultimate aim is that after this whole cascade, the last protein or transcription factors, it goes inside the nucleus and it causes the transcription. So in this way, this signaling works. So that means one ligand, the ultimate goal of this ligand was to cause the transcription of this particular transcription factor. But it cannot cause the transcription of this transcription factor directly. So it has to go through a signaling cascade. And this signaling cascade ultimately causes the transcription of that particular transcription factor that it is aimed to. Okay, so this is essentially a paracrine signal. I hope it is clear, clear to you, clear to everybody. Okay. Now we'll go to the next question. So why I wanted to discuss this, because it's important, these are very basic concepts. So no matter whether we are solving 100 questions, 10 questions, or 15 questions, but like I always say, your concepts should be clear. These are very basics of how a path signaling pathway works, and what is that like. You raised a very good question, that is it similar to the antigen antibody interaction? No, although both are proteins only, but that interaction is a different kind of interaction. After your antigen antibody interaction, your antigen is endocytosed and it is broken into fragments, either by MHC molecules or in a MHC dependent manner, or it is degraded by your neutrophils or some other natural killer cells. So that is a different mechanism. But here, the in antigen antibody interaction, your ultimate goal is to destroy the antigen. But here, your ultimate goal is not to destroy the ligand. The ultimate goal is to activate a certain transcription factor, because ligand cannot directly activate that transcription factor. So it has to go through a cascade. For example, if I want to do graduation, I have to go like from first, second, third, fourth. Then I have to study up till 12th. Then only I can take admission in my BSc or BTech. I cannot take admission directly from nursery to BTech. I have to go to the series of cascade of classes. I have to complete all the classes. Similarly, here also, that a ligand, ultimate goal of the ligand is to activate a transcription factor, but it cannot go directly. It has to go through a signaling cascade. And those signaling cascades in developmental biology are of different types. The first one is RTK that we have just read or see. There are many other like uh, Wnt pathway, wingless pathway, or TGF beta pathway, and all of those things. Fine. Now, next question is easy question. Nothing much concept to be explained here. Match the following. So FGF1, FGF8, FGF2, FGF7, limb development, blood vessel formation, skill development, regeneration. So you have to do the matching. I'm giving you 3 minutes for this question. Think and answer. Okay. Think and answer. Hmm. 1 minute more and then I will switch the screen. Okay, now I'm switching the screen. Let's see. Mm. Okay. So, no, I'm not fully recovered. I'm still suffering from cough and cold. I don't know why it's not going. It's more than 15 days now, but it's still like this. Still, I have to take class. Anyways, this is the question from sixth week. No, this is not the question from sixth week. I don't know whether this question, if this question is given in your assignments, I don't know. But this topic was there in your fifth week. This thing. So yeah. So Ajay has answered AP. That is FGF1 in skin development. Okay. BM. BM. FGF8 in limb development. CN. CN. Hmm. Okay. So the correct answer is this one, the first one. And so this is the table which is given in your fifth week this thing lecture. Nothing much to explain. So this is the fibroblast. FGF stands for fibroblast growth factors. Okay. So FGF1 is responsible for regeneration. FGF2 is responsible for blood vessel formation. FGF7 is responsible for skin development. And FGF8 is responsible for limb development and lens induction. This is the table which is, I mean, this is the text which is given. On the basis of this, if you see, the option would be the first one. Okay. So just have a look over here. From this, they have framed the question. Easy question, nothing to explain. It's just to like a memorizing kind of a question. You just memorize the data. Okay. Now we'll come to the next one. It's a big question, and it's an interesting one. Hmm. In an experiment, muscle, brain, dermal, and intestinal tissues were enzymatically dissociated. Okay. Then myocytes, neurons, hypodermis, and the lumen epithelium of the intestine were isolated separately and mixed. Okay. Now, based on Steinberg's hypothesis, identify the correct order of cells from the outside to the inside in the aggregated mass. So these are your options. Now think. It will take time. I'm just reading the options. A is neurons, hypodermis, myocytes, luminal epithelium of the intestine. B is neuron, myocytes, hypodermis, and luminal epithelium of the intestine. C is hypodermis, myocytes, neurons, and luminal epithelium of the intestine. D is hypodermis, neurons, myocytes, and luminal epithelium. E is luminal epithelium, hypodermis, neurons, and myocytes. And F is luminal epithelium, neurons, hypodermis, and myocytes. I'll give you 3 minutes for this question. Think and try to answer this. It's 6:55. 6:58. I will switch to the screen, okay? Try to answer this question. Try to think what is asked. Take your time. think, see how first you have to see what is Steinberg's hypothesis and how this Steinberg's hypothesis can be implied here. Okay. One more minute left. Okay. Now I'm switching the screen. Hmm. Fine. Okay. So, hmm. So Ajay has answered C. That is hypodermis, neuron, myocytes, and illuminal epithelium. Yes, it is the correct answer. Ecto, meso, and endo. Can you explain this question? Otherwise, I'm going to explain. Can you explain how you solved this question, Ajay?
Madam, first I had divided it into ecto, meso, and endo. And then N cadres, they have more higher surface tension than E cadres. That's why E cadres has to be outside and N cadres is to be inside. That's why hypodermis above, then neuron, then myocytes, then luminal, and then luminal epithelium of intestine. That's what logic. I don't know whether it is correct or not. Means my logic is correct or not. I don't know.
The logic is correct. You have answered it correctly. Very good. So basically, this question says that based on the Steinberg's hypothesis, identify the correct order of cells from outside to inside. So first you have to answer the one which is outside, and then you have to go up to the inside. Fine. So Ajay has answered uh, uh, C. And let's see whether C is the correct answer or not. Yes, C is the correct answer. So first you need to know what is Steinberg's hypothesis. Okay. So basically Steinberg's hypothesis is a differential adhesion hypothesis. Differential adhesion hypothesis. Just a minute. So Steinberg's hypothesis is differential differential addition hypothesis, which explains that how differences in the intensity of cell adhesion, it can give rise to spatial interactions. Means differences in the intensity of cell adhesion, that means that cells can adhere to themselves, they have an adhesion property, but not all the cells have the same adhesion property. There is a difference in the cell adhesion property of different cells, and this is exploited, and therefore there are different spatial interactions. That means cells which are present in the interior, in the outside, so they have a higher adhesion property, they are more adhere to each other. Whereas cells which are forming the outer layering, the outer covering, they might have a lesser adhesion property. Now, what determines their adhesion? The adhesion is determined by what? It is determined by what is the thing that determines the adhesion?
Calcium adhesions. They are
Calcium
called
adhesins.
cadres.
Yes, which is known as cadherins. So there are different kinds of cadherins like N cadherin, E cadherin, and these determine the adhesion adhesiveness. So these are cadherins are actually calcium dependent addition molecules which are responsible for the interactions in cell to cell additions. Now this is one thing. Now how Steinberg's hypothesis comes into role here? So you need to understand that relative amount of cadherin matters. How? Because Steinberg's hypothesis says that uh, all those uh, uh, all those structures or all those cells which has a higher surface tension, they are present in the central location of the embryo. Steinberg's hypothesis, it explains the spatial arrangement on the basis of surface tension. Okay, it explains. Just a minute. Hmm. It explains spatial arrangement on the basis of surface tension. What does it mean? It means that all those tissues which has a higher surface tension, they are located in a central location in the embryo. So higher your surface tension, they have a more central location in the embryo. Higher surface tension, they have more central location in the embryo. Okay, that means different cells have different surface tension properties, and the ones that has a higher surface tension, they will be in the center. Now what is the meaning of spatial arrangement? Spatial arrangement means that in 3D space, in a three-dimensional space, how your cells are arranged. So if you have a circle is a 2D representation, and a sphere like a football, it's a three-dimensional representation. So in a three-dimensional representation, how your cells are arranged. So in your three-dimensional representation, some cells will be in the center, some cells will be at the periphery, some cells will be in between your periphery and the center. Okay, if this is your, suppose if this is your three dimension circle, so some cells would be here, some would be here, some would be in between. So what decides that some cells should be in the center, some cells should be in the periphery, and some cells should be in between? So to explain this, a uh, theory was given by Steinberg, which says that all those cells which have a higher surface tension, they are present in the interior, on the central part, and all those which has a lesser surface tension, which are loosely attached, are present in the exterior parts. So this was a theory which was okay. This was the theory which was given by him. Now this is clear. I think this should be clear to you. Now, what is the role of cadherins over here? Now we have to link the role of cadherins with it. So important is that more cadherin, higher the surface tension and more central location it will have, or also the kind of cadherin it takes, that also decides. So cadherins are responsible for the adhesion properties. So if you have more cadherin, that means the adhesion is more strong. I mean, adhesion is stronger. If the adhesion is stronger, it will take the central location. All these concepts are very similar. You know, we say biology doesn't differentiate in chemistry and zoology and botany. So if you remember, we talk about the structure of atom. There also we talk about that the electrons which are in the outer shell, they are very loosely packed, and the electrons which are inside are very densely packed. Same similar kind of concept applies here also. that all the cells which are in the interior, because they have a greater amount of cadherin, so their adhesion is very strong. Whereas cells which are, or tissues which are at the outer, they have a lesser surface tension because the cadherin is also less, and therefore their location is in the periphery. Okay, if sometime later I will also explain it in the in your molecular biology terms, in molecular biology terms also. Just a minute. Okay, excuse me. Yeah, so in molecular biology terms also this holds true, that genes which are very much expressed, whether it's chemistry or developmental biology or on the molecular level, it is all the same. Nasreen, do you want to say anything? Uh, if there are no questions, then you can mute yourself.
No voice, madam. Your voice not coming.
Hmm, okay, fine. So if you, Nasreen, your voice is not coming, if you want, if you have any question, you can just write it in the message box, okay? And I will request everybody to kindly mute, because that might create some problems and disturbances in the class. So if you are audible, if you can hear me, then please mute yourself. If you have a question, you can raise your hand. Okay. Anyways, fine. So, like I explained, so this concept also applies in molecular biology. In molecular biology, at the atomic, at the nucleus level, genes which are highly expressed are present in the center, the euchromatin part, and genes which are not expressed in that particular cell, the heterochromatin part, they are present in the periphery. So what I'm trying to explain here is that spatial organization of cells and tissues is very important. In biology, always remember that spatial organization, whether it is the spatial organization of your cells or tissues, whether it is the spatial organization of your atoms in different orbitals, or whether it is a spatial organization of your uh, DNA, I mean, euchromatin or heterochromatin part in the nucleus. But spatial organization plays a very important role. And in this question, if you can see, so in this question, here we are talking about the spatial organization of the cadherins. So cadherins, because the regions uh, uh, which has a higher surface tension, that means it has more cadherin, and they are present in a more central location. Okay, so if you see hypodermis, myocytes, these are what? These are the hypodermis and myocytes. What are these? These are the ectodermal cells. Okay, and then your luminal epithelium of the intestine, what is this? It is an endodermal. So that means if they are endodermal, that means what? They are endodermal, that means they have a higher surface tension, that's why they are present inside, no? And that means their cadherin level would also be high. So it's a very easy question. If you see, based on, even if you do not know the Steinberg's hypothesis, by just knowing whether it is an ectodermal cell or endodermal cell, you can solve this question. That obviously the ectodermal cells would be present in the outside region, and the endodermal will be present inside. And since luminal epithelium is the endodermal, so obviously it will be present inside. Hypodermis, myocyte, what are these? These are ectodermal, so they will be present outside. So just simply by knowing which is an ectodermal cell, which is an endodermal cell, also you can solve this question. Okay, but all these. Yes, Ajay. Yes, what is your query? Okay, just a minute. Yeah. So your question is hypodermis is different from dermis, since dermis is from uh, mesoderm. Yes. So dermis is basically the middle layer. Okay. And first is your epidermis. No, hypodermis and dermis are, I don't think so that there is any difference, because epidermis is the top layer, and then after epidermis you have your dermis, which is the middle layer. Okay. So hypodermis, just a minute, I also have to check this.
Because I never heard something like hypodermis. Hypo means it should be below the dermis, na. Skin has
What
only
is
two layers, epidermis and dermis.
Yeah. So
Hypodermis will be muscle only.
So what I could understand is that first you have your uh, epidermis, okay? Beneath your epidermis is your dermis, and beneath your dermis is your hypodermis. Okay, so first you have your dermis layer, then you have your epidermis layer, and beneath your epidermis you have your hypodermis layer. This is the correct order, I think. And basically, your hypodermis, what I could understand is basically the area which has all the fat tissues or the connective tissues. Okay, and it also has some kinds of maybe follicular tissues can also be there. So, although this is not a very general term, general notion, but all you can understand is it is the bottom layer of the skin. And mostly your the functions of hypodermis is storing your energy, connecting your dermis to your skin, and protecting your body. So basically, what the kind of insulation that you get in the body, that you get from the hypodermis, because the fat layer, the fat skin, it is in the hypodermis. And that is why uh, what I could understand, like what I am also reading, but I think what I could understand is it says that as you age, your hypodermis decreases in size, and that is why your skin starts sagging. Your skin starts loosening up. So I think the elasticity property and the tightness property of your skin, it also depends on hypodermis. Because as we start aging, the hypodermis it starts decreasing. I mean, it starts decreasing its size. This is what I could understand by reading right now. So you can, I
So
think you can all,
confusion is more, madam. Now, hypodermis is it coming from ectoderm or mesoderm? It's coming
what
from
I could
mesoderm then.
see, what I could learn from this is that it is coming from it should come from mesoderm only, because it has connective tissues, and whatever connective tissues that we have learned, like blood vessels and all, they form the regions of mesoderm. Mesoderm only has the connective tissues. But when we talk about skin, skin has epidermis, dermis, and hypodermis. And hypodermis is the lowest, that means the bottom of the bottom layer of the skin. Okay.
Madam, still, I still have problem with the framing of the
Hypo,
question. It is not
see,
really good.
hypodermis
I
comes from
actually
mesoderm.
fail to understand. They are in a difficult English, I think.
Sorry, I could not understand what you're trying to say. Can you repeat the question?
Framing of the questions is not easy, madam, in developmental biology. They
Yeah, I
are
know. I can understand this, that the framing
I
can be a
request
little.
them that they should be in easy English. Means see how much confusion the hypodermis is. Rather than should have given ectoderm or epidermis. That would have been better.
Yeah, I know. But just try to understand that hypodermis is also derived from mesoderm, but unlike the dermis layer, it is derived from some other region of the mesoderm. It is not derived from the dermatome region of the mesoderm. Okay, so hypodermis also comes in mesoderm only. But Normal dermis layer is derived from the dermatome region of the mesoderm. It is not derived from dermatome region. Don't go into so much detail. Just remember that your skin has three layers: epidermis, dermis, and hypodermis. And hypodermis is derived from the mesoderm layer. Okay. And about the framing of the questions, that I mean, if you have like a one-to-one session with the course instructor, then there I think you can put your this thing query or put your suggestions there. Anyways, now next question is: an embryo at an early organogenesis stage was dissociated and subjected to Western blot. Based on your understanding of the hierarchy of interactions, identify the specific gatherings from the given blot that are precursor of their respective tissues. Okay, this is a little tricky question. So you have to tell that these are the, it is an image of a Western blot. Okay, now you have to tell that what does these lanes, the bands of these lanes represents, whether A is retinal pigment epithelium or secretory cells of thyroid, smooth muscles, skin. So you have to tell that which lane represents which band. So read this question as many times as you want. I'm giving you 3 minutes to answer this question. Think and try
Same
to
logic
answer.
can be applied here. It's very easy, I think.
It's easy, but let's uh, wait for everybody, and everybody should get an equal time and opportunity to answer the question. Okay, think, try to understand the question first. Try to understand the language of the question. One minute more. So whoever is answering the question, try to explain also. Okay, now I'm switching the screen. Hmm. Okay.
Madam, same logic I applied last time. I don't understand the charting. Outermost is skin, then retina, the smooth muscle, and then thyroid. And same thing they appear on the chart. Only C is upright. I don't know. In the
Okay. Hmm. Anybody else who want to try? Sangeeta, Jasreena, Nasreen, Akhila, Nandana. Anybody who want to try? Think. Try to answer. We have solved a similar question just now. You can try to apply some of the concepts. So cadherins level would be high in the muscles or in the cells which are present in the interior. Okay. So you have smooth muscles, you have skin, and you have some thyroid cells. You know which are, so skin is present in the interior or exterior. Thyroid cells would be present in interior or exterior. Think with that logic, you will be able to answer this question. Just now we discussed now. Think what can be the answer? Hmm? Sangeeta, can you think of something? Akhila,
Ma'am, the skin will be the last. Skin will be the last, and
last means
because
can
it's
you
outside.
explain in with this? I mean this image. Let me just.
Yeah, in this image, I think this one, the smooth muscle, then secretory cells of the thyroid, then retinal pigmented epithelium, and the last is the skin. If I'm right.
Okay. So which option are you choosing? Can you tell me please from these?
Uh, option is C, B, A, and D.
Okay, you are saying C, B, A, and D. Let me just write it here. C, B, A, and D. Is there any options? But there is. I don't see any option like this. C is retinal pigment.
C
Okay.
is
Okay. At least you have tried. It's a very good thing that at least you have tried. So, the correct answer is skin A. Okay. Let me just write it somewhere. A, B, C, and D. Skin, retinal pigmented epithelium, smooth muscle, and secretory cells of thyroid. So you see here, secretory cells of thyroid, which what are they? They are ectoderm, endoderm, or mesoderm. What are they? Anybody?
endoderm,
Endoderm. Smooth muscles,
mesoderm,
mesoderm, and skin,
no,
ectoderm. So,
ectoderm.
so that means it is going like ectoderm, then mesoderm, and then endoderm. Now, skin, smooth muscles, retin. Okay, so this is the second last option, right? Hmm. This option is correct. Now the question says an embryo at an early organogenesis stage was dissociated. So if you have an embryo, okay, now it was in a very early organogenesis state, means it was not completely made, it was making its organ. So in the early organogenesis state, it was dissociated and subjected to Western blot. Subjected to Western blot means Western blot, what does it tells? It tells about the protein levels, not exactly levels, but whether that particular protein is present or not. If the protein is present, specific protein is present, it will give a band. If a specific protein is not present, it will not give a band. Okay, I am explaining from the scratch because I want everybody in the class to understand. So uh so I think someone have, you can mute yourself, whoever has unmuted. Good, fine, thank you. So This is an embryo which is in an early organogenesis stage, and it was dissociated. Dissociated means it was broken into pieces. It was completely opened up and subjected to Western blot. What is the meaning of Western blotting? Western blotting means to detect whether protein is present or not. So we have a we can do it. We have a when we do electrophoresis, we can have a gross gel electrophoresis or a SDS page or normal page electrophoresis, and we can have a northern blotting, and we can have a western blotting. So, and we can have a southern blotting also. So when we talk about western blot, we are specifically talking about proteins. Remember this. And what is southern blot? Can anybody tell me what is a southern blot? Southern blot is for
DNA
DNA.
and
Southern blot
RNA.
is
DNA and
and
RNA get separated.
northern blot is for
Northern
northern
blot for
blot is
RNA.
for RNA. So we have three kinds of blot. We have a southern blot, we have a western blot, and we have a northern blot. Okay. Southern blot is for DNA, western blot is for proteins, and northern blot is for RNA. Okay. So from this question, one thing we could understand that it is subjected to proteins. Means the proteins are the proteins were taken out. Now, based on your understanding of the hierarchy of interactions, identify the specific cadherins from the given blot that are precursor to their respective tissues. Okay. Now, the specific cadherins is not something, the language is a little bit difficult, but you just try to understand that you see here, first lane would be skin. First thing that you can identify, which is at the very exterior, is the skin. Then you have your epithelium layer, then you have your smooth muscles, and then you have your endodermal cells, which are the cells of the thyroid. So ectoderm, mesoderm, and endoderm. Like this, you are going. So just you have to understand that the levels of cadherins would be less when it is an ectoderm. Okay, you can see here. See which has the most amount of cadherin level, that would be the endodermal part. Okay, so here D has the most amount of cadherins, so it would be the endodermal part. Which has the least amount of cadherin, it will be the outermost. So this has the least amount of cadherin, so it will be the outermost. So now see, two things you have to see. First, you know that the sequence would be that in the innermost part it has to be endoderm, the outermost part has to be the skin, and then after that smooth muscles, after that retinal pigment. This is something that we already know. Now we have to correlate it with the cadherin level. Could you understand? First, you should know on the basis of your ectodermal and endodermal arrangement. First, you should know the sequence. The sequence would be skin, smooth muscle, retinal pigmented epithelium. Sorry, skin, retinal pigmented epithelium, smooth muscles, and secretory cells. Now you have to correlate with the cadherin levels. So skin has to be at the first place, and you can see since skin is the outermost layer, so it will have the least amount of cadherins. Endodermal is the innermost layer, so it will have the most amount of cadherin. Then you have retinal pigmented epithelium, and then you have smooth muscles. So in this way you can try to correlate it with the with the cadherin levels also. Jasrina has also tried to answer. Good. Okay, now we'll come to the next question. Okay, again a little bit longer
Madam, what for
question.
that L is there? Sorry, I didn't understand anything from the chart. What is
Actually,
that
this chart is a
names were
little bit
given?
confusing.
LA, KA, DA. Means the darkness shows more amount or the number specific shows more amount.
L is the ladder. Okay, this is the ladder.
Okay.
This is the,
And 10 is for 10, 55, 75, 180. These are the kilodaltons.
these are the size of the proteins that have been determined. So this protein, I mean this particular band is somewhere between 10, 20, 30, 35. Let's, I don't know what is the ladder, like what this particular band denotes, but just assume that your this band is between 10 kilodalton and 35 kilodalton, or if it is 20, then 20 kilodalton, whatever this ladder says. This band is somewhere uh, below 35 kilodalton. So this is the
Actually,
size of
madam,
the band.
the
See,
sequence should have been A skin, B retinal pigment,
hmm,
C then C
smooth
should
muscles.
be secretory cell of thyroid if they are endoderm, and D smooth muscle.
Can you repeat what you're saying?
Instead of skin, retinal pigment, smooth muscle, and secretory cell of thyroid, the
Mm-hmm.
sequence of C and D should have been reversed. Means C should have been secretory cell of thyroid, and D should be smooth muscle. Then it would have been means according to the surface theory.
Hmm.
Means first
I
skin, then
understand.
second
Hmm.
retinal pigment, third we should have got smooth muscle, and highest will be that of secretory cell of thyroid.
Hmm.
But that option is not given here.
Yes, I know that option is not given. So what I could understand from the question is, if you know that in Western blot there are two things that need to be considered. First is the size, and second is the band size in accordance with the ladder, and second is the band size. So if you have a band like this, and if you have a band,
Band of B is more than C.
that is a little confusing here, that if we see the band size, then the band of B is more than C. But you see, both the things have to be considered. See, the band of B is more than C, but the difference in the band size is not that much as compared to the difference in their uh, protein dalton size. Could you see here? This is a 180 kilodalton protein, and it is a 35 something below 35 kilodalton protein. So this question is a little confusing, because either they should have mentioned to consider the band width, or they should have told to consider the ladder size, because here we have to take in accordance both the things, the band width also and size also, the size in comparison to the ladder, because that is why they have given ladder. Otherwise they could have only given the band width. Because they have given the ladder, which means that we have to take into accordance the band width also, and comparing it with the ladder also. So both things we have to simultaneously see to answer this question. It is not just that we have to consider the bandwidth. Both things we have to consider. So it's a confusing question, because three things are there. Whether they want to ask whether the what is the sequence, or they want to compare us with this graph in accordance with the band size, this band size, or they want to consider in accordance with this bandwidth. So there are three different contrary things are there. But as the answer is, you can see it is the smooth muscles are the second last. So with this, what I could only understand is that why they have given smooth muscles in the second last is because the size difference. So this is a confusing question, I agree. Only thing, two
Sorry,
things only
ma'am.
you can
Though
understand is,
my answer is correct, I am not satisfied.
then it's fine. If you're not satisfied, then you can like whenever you have a, as I said earlier also, you have a live interaction session, you must have a live interaction session. I think they will be conducting after half of the this thing is done. Might be after the next this thing live interaction session, you might have a session with your course coordinator as well. There you can ask these whatever doubts you have. Okay, that why they have given this answer as correct option and why they haven't given. So these little bit queries you can ask them. So this, it is not in my hand, it is not in my control, so I cannot do anything on that. Anyways, so now we'll come to the next question. Cadherin mediated cell to cell contact induces downstream signaling via phosphorylation of amino acid residues in the cytosolic domain. In an experiment, antisense oligonucleotides were introduced in the cell, which resulted in the loss of cytoskeleton response to cadherin binding. Further experiments revealed that there was no change in cadherin recognition or downstream phosphorylation. Okay. The loss of response was due to the lack of coordination between the extracellular matrix and cytosolic components. Fine. Based on this information, what could have been the target of these antisense oligonucleotides? A. Alpha actin. B. Beta actin. C. Calcium binding EC2 domain. D. Alpha catenin. And E. Beta catenin. Think. 3 minutes for this question. It's 7:38 right now. 7:41. I'll switch the screen. Okay. Try to understand the question first. So one thing, if you have questions like this, then you have a very lengthy question, because when you are doing, you're giving your CSI, when you will give your CSI exams, you will have such kinds of lengthy questions, paragraph kinds questions. So don't get panic when you get such paragraph kind of questions. Try to, you can just divide the question into smaller bits and try to understand the meaning of each and every line. You can draw it, you can write it on a piece of paper. That can help you to understand the question. Okay. So I am telling it for everybody in the class, because we have to take everybody together when we are doing a live session or any kind of class. So for questions which are so big, try to understand the meaning of each and every sentence. Break your question into smaller pieces and then try to correlate each and every line with each other. Okay? Now try to solve this question. Think. One minute more. I'm giving you. Okay, now I'm switching the screen. Okay, fine. Let's see. So all these questions are actually very conceptual questions, like not direct questions. But that's fine. To have conceptual questions is fine. Only thing is that you need to understand the language. Hmm. Okay. Okay. Good. So students have answered it. So Ajay has answered D and E, that is alpha and beta catenin. And Nasreen has answered as beta catenin, and Jasreena has also answered as beta catenin. So alpha and beta catenin are our two majorly answered options. And the answer is alpha catenin and beta catenin both. Good. So that means you could have you have understood the question. The question says that cadherin mediated cell to cell contact induces downstream signaling via phosphorylation of amino acid residues in the cytosolic domains. Yes. Then in an experiment, antisense oligonucleotides were introduced in the cell, which resulted in the loss of cytoskeleton response to cadherin binding. Further experiments revealed that there was no change in the cadherin recognition or downstream phosphorylation. So this line is important. Just a minute. Here it says, I have made this figure, matlab, taken this figure. Hmm. The question says that there was no change in cadherin recognition. So if you see that this is particularly the way in which your cell to cell communication takes place. Here it says antisense oligonucleotides were introduced. So antisense oligonucleotides were introduced. This means that something is getting blocked, because the function of antisense RNA or antisense miRNAs is to block the transcription or to block the expression. So antisense oligonucleotides were introduced in the cell, which resulted in the loss of cytoskeleton response to cadherin binding. So when the cadherins, it binds to the cytoskeleton, but the binding of this cadherin to cytoskeleton was blocked. Okay, so these is the cytoskeleton and these are your cadherins. So the binding of cadherin to the cytoskeleton was blocked. And how it was blocked, we don't know. It says that further experiments revealed that there was no change in cadherin recognition or downstream phosphorylation. That means these are the recognition sites. So cadherin recognition was fine and phosphorylation sites was also fine, but still somehow the the response of this cadherin to the cytoskeleton was not made. Then the loss of response was due to the lack of coordination between the ECM and the cytosolic components. Based on this information, what could have been the target of these antisense oligonucleotides? That means what are the things that are responsible for this interaction? Your cadherins, the recognition, phosphorylation, and catenins. It is already mentioned in the question that the recognition is fine, phosphorylation is also fine. Then what is left? Your catenins are left. That means the antisense has targeted your catenins. That is why the recognition or the interaction of the cadherins to the cytoskeleton is blocked. Okay, so I have explained you with the help of this figure. Just a minute. Is there any question? Okay, no. So Aditya, welcome to the class. You have joined in a very correct time when there is just 10 minutes left for the class. Welcome. Why you have joined so late? I mean, I'm not saying to leave the class, but I just wanted to know the reason why you have joined so late.
Sorry, ma'am, I was not well.
Okay. So now are you fine?
Not exactly, but I thought what you were taking, I could get an idea.
Okay. So anyways, if you're not well, there will be a recorded session, so you can go through it and just have a look of what we have discussed. So we were discussing a question. Um, Yeah. So we were discussing this question. So here, I think I'm not going to repeat it again because we still have some questions and we just have 10 minutes left. So I think now this question would be clear, although most of you have answered it correctly. Based on this figure, we can see that since this is also correct, this is also fine, there is no problem here also, there is no problem here also. That means antisense has targeted these catenins. Okay, fine. Now we come to the next question. Hmm. I think we have two more questions left. We'll be able to solve it in the time. Yeah. Which of the following conditions will completely abolish the invagination of ventral furrow in Drosophila? RNA interference against twist and a gain of function mutation in row A. RNA interference against twist and snail. Gain of function mutation in twist and RNA interference against row A. Gain of function mutation in row A. RNA interference against beta catenin alone. RNA interference against row A alone. 2 minutes. It's 7:48. 7:50. I will switch the screen. Okay. 1 minute left. So I'm switching the screen now. Hmm. Fine. So Ajay has answered as B, C, E, and F. Let us see what are these options. B is RNA interference against twist and cell. Okay. C is gain of function in twist and RNA interference against row A. Okay. B, C, E. RNA interference against beta catenin. Okay. And F. Right. So this is the correct answer, I think. Let me just confirm. It is, yeah, it is correct. So can you explain me why you chose these options? Ajay, can you explain this question to the class? You'll save my energy also.
For formation of ventral furrow, twist and snails, these two types of caterins are required. And for branching, B and L, S, third is required. Okay. So if something is acting against twist and snail, so it will abolish the ventral furrow.
Hmm. Yes.
Then gain of function mutation in twist and RNA against row. Row A is important for formation. So again, it will there. And beta
Yes.
catenin alone or row alone, the same reason is for E and F.
Yes.
Because row A, twist and snail, they are important for formation of ventral furrow, which invaginate and form the mesodermal layers.
Hmm. Yes. So he has answered is correctly good. So basically, you should understand that whenever we are talking about ventral furrow in Drosophila, twist, snail, and row, these three are very important. Okay. So basically, twist and snail, these are the transcription factors, and they will try kind of initiate the ventral furrow formation. Okay. Twist and snail, they will initiate the ventral furrow formation, and ultimately the ultimate factor which is responsible for regulation of the cytoskeleton dynamics and ultimately the ventral furrow formation is your row A. So if we want to completely abolish the invagination, here we are saying how we can completely abolish the invagination. So if we have to completely abolish the invagination, we have to disrupt the factors which helps in the imagination process, and these factors are what? Twist, snail, and your row A. So twist, RNA interference against twist and snail. So RNA interference will stop the transcription of twist and nail. Okay. Gain of function mutation in twist and RNA interference against row A. So ultimately, you understand that ultimately the guiding factor for the invagination is row A. So even if there is a gain of function mutation in twist, but because there is an blockage in your row A, then it would be ventral furrow would be stopped. Gain of function in row A, no, because if there is a gain of function in row A, then there would be some other abnormality, but ventral furrow will not stop. It might grow even more. RNA interference against beta catenin alone, yes, and RNA interference against row A alone, yes. So basically, you need to understand that. Okay, so just a minute. Actually, I'm speaking continuously since 2 hours. My throat is also not very good. So, twist, snail, and row A. These three are important for your ventral furrow invagination. So, if we are blocking these, then the abolishment of the ventral furrow invagination will take place. First thing. Second thing is most important factor and ultimate guiding factor for the ventral furrow formation is your row A, because row A controls the cytoskeleton dynamics. Okay, this row A is basically, what is row A? RAS row. It's actually a small GTPs. Okay, so if you know, you have RAS, RAF, row. These are GTPas, GTP activating proteins. So it is basically a small GTPas which regulates your actin cytoskeleton dynamics. So I think this much explanation for this particular is enough. You can go and you can read about the exact mechanism of how twist, snail, and row control it. You will get to know that why row is more important. Anyways, now we come to the next and the last question for the class. I hope it's the last one. Yeah. Which of the following are a consequence of cadherin depletion? Impaired melanocyte migration, impaired CNS development, impaired notochord formation, failure in segregating neural plate and epithelium, invagilation of neurola but failure to form melanocytes, conversion of neural epithelium to epidermis. 2 minutes. It's 7:55. 2 minutes. 7:57. I will switch the screen. Think what can be the answer. Okay, I'm switching the screen now. Mm. Okay, so Ajay has answered A, B, C, D, and Jasrina has answered B and C. Fine. So one we have A, B, C, D. A, B, C, D. Okay. A, B, C, D, and also we have as B and C. Hmm. So what is A? Impaired melanocyte migration. Okay. B is impaired CNS development. Okay. Then C is impaired notochord. Okay. D is failure in segregating neural plate and epithelium. Okay. And invagination of neurula with but failure to form melanocytes. I don't think this is the answer. And conversion of neural plate. Okay, I think you have answered correctly. Yeah. So A, B, C, D are the correct answers. So Justina has also answered two of the options as correctly also. So good job. So basically you need to understand for this question, you just need to know what is the function of N-cadherin. So N-cadherin, as we know, it's a cell adhesion molecule and it is involved in developmental process, particularly involving nervous system and tissue segregation. So N-cadherin is involved in processes which involves nervous system and tissue segregation. So if it involves nervous system, it also involves some kinds of melanocyte development also. And cadherin is also involved in melanocytes. That is why you will see that N-cadherin has role in some kinds of skin cancers also. So from this nervous system, tissue segregation and melanocytes development regulation, you can see that when N-cadherin is depleted, you will have impaired melanocyte, impaired central nervous system, impaired notochord, and failure in segregating neural plate and epithelium. Because these are involved in nervous system, this is involved in segregation, this is your melanocyte. So just you need to know what are the functions of N-cadherin. Okay. So with this, we finished today's class. So today in this class, we have dealt the questions which are related to cell communication. And in the next class also, I think we'll be discussing about cell to cell communication and we'll be discussing more about the pathways which are responsible in your this thing in your developmental biology. Like this time, if you have gone through the lectures, there was receptor tyrosine pathway. You have other pathways also like TGA beta pathway, wingless pathway. Those questions related to those pathways will be dealt in the next Tuesday's class. So if you could not follow up or if you were late due to any reasons, so you can go through my YouTube channel, which is Apuba Sony NPTEL TA. You can type it, you will get it. So all the recorded sessions are available there. And you might also have a link where you can find the PPTs that we discuss in the class. Okay. So thank you everybody and a very happy Janmashtami to everybody who is celebrating today.
Thank you, madam. Do try that acetylcysteine, madam. That is
Yeah,
helpful.
yeah, yeah. I'll take it. Thank you. Thank you so much.
Mm.